Hello everyone. In this video I'm going to speak about real-time PCR. Of course there was a previous video about the basic principle of PCR. So if you don't know anything about PCR, I highly recommend you to go there, watch the previous video about PCR and then come back because then it will be much easier for you to understand this video. The link of the PCR video is in the description of this video, so you can go there directly. Now, in this video, I'm going to speak about real-time PCR, the principle, the technique, how does it go, and everything related. I also will speak about reverse transcription because it's used widely in real-time PCR, and also the data of real-time PCR. So as usual, let's start by asking what is real-time PCR. Real-time PCR is basically a modified technique from PCR. So what's the difference? As I told you in the previous video, PCR is a thermocycler. So it performs a thermocycle by heating up and cooling down the temperature in 3 steps, which are the DNA naturation, then primers annealing, and then the DNA extension. Which these 3 steps represent one PCR cycle. And then after each cycle, we will get double the number of PCR sequence we have. So if you have first 2 sequences, then we will get 4, then 8, and so on. So it's a process of DNA amplifying, an amplifying of a specific DNA sequence or gene. Now, of course, in PCR, what we can do is to set the time. We can set the time for 1 hour, half an hour, or so on. And at the end of the PCR process, we can, of course, we can get our sample and we can measure the amount of DNA we have at the end of the PCR. But we will never know how much PCR we had during the cycle. And this is the modification in the real-time PCR, because real-time PCR is a combination between PCR and spectrophlorimetry. So it's like normal PCR, but it's engaged with spectrophlorometer, which can measure the amount of DNA in my sample continuously during the PCR process or after each cycle. So I will get an estimation how much DNA I had in each cycle of my PCR process. So let's say this is the PCR. We have double numbers of DNA sequences after each cycle. The spectrophlorometer will be measuring the amount of DNA in my sample during the PCR continuously. And this is how does it work? So this is a diagram of the real-time PCR machine. So here is what we call the tube patch. It looks like this from outside. So here is when we put our tubes. Normally we use plates because we can apply real-time PCR to several samples at one time. So we can use the 96 Wells plate or whatever. So here is the thermocycler, like the normal PCR. This what you're seeing here is the normal thermocycler. And what you're seeing here is the extra, which is the spectrophlorometer. So the thermocycler undergo the normal thermocycle, the 3 steps thermocycle. And then the spectrophlorometer measures, so this is how the spectrophlorometer works. So it excite a specific fluorophore inside the sample, and then the fluorophore emits fluorescent or emits light, and we then detect the light coming out of the sample. So here is how the spectrophlorometer works. So the spectrophlorometer keeps measuring the amount of DNA in my sample continuously during the PCR process. Because of this, it's called real time. Here is how the spectrophlorometer works, basically. So this is the light source. Then we have mirror. We have the monochromator to choose like a specific wavelength. Then the light causes an excitation to the fluorophore, which emits light, and we can detect in the detector. So this is the spectrofluorometer, which we see here, and this is how it works. So this is the real-time PCR actually. In real-time PCR, it's like normal PCR, but we are measuring the amount of DNA we have inside the sample continuously during the PCR. Because of this, it's called real-time. Now I'm going to speak about how does it work? How does real-time PCR works? As I told you previously in the PCR video, that we need 4 essential elements in order to perform a PCR process, which is first we need the parent DNA, we need the DNA sequence or the specific gene we want to amplify. Then we need the DNA contents because we need to synthesize DNA. So we need the DNTPs, which are the deoxyribose nucleotide 3-phosphate, the adenine, thymine, cytosine, guanine, the DNA content. We also need primer. We need a couple of primers, the reverse and the forward. This I told you in the previous video, and of course we need the very important enzyme, the DNA polymerase, the heat-resistant DNA polymerase to produce DNA. Now in real-time PCR, we need one extra molecule, which is very important, and which is the detector. The detector is the thing we use to detect. The detector is what the spectrofluorometer detects. So this is the very important thing in the real-time PCR, the detector. So I'm going to speak about the detectors used in the real-time PCR. We have 2 types of detectors, the non-specific ones and the specific detectors. The non-specific detectors are fluorescent dyes. The most known of them is the cybergreen. The cybergreen is a molecule, is a fluorescent dye. It looks like this, and I'm going to speak about it in details. The specific detection is a detective probe. It's a probe which is very specific to the gene or the specific sequence I want to amplify. The most known is the TACMAN, and it looks like this. It's a probe, and it's very specific to the gene or DNA sequence I want to amplify. So I'm going also to explain this in details. So let's first speak about cybergreen. Cybergreen it's a green fluorescent, of course, fluorophore. From its name, it's green, and it can so it can realize or it can detect the double-stranded DNA. It's very sensitive to double-stranded DNA. So let's say we have a double-stranded DNA. The cybergreen can bind to the minor groove of the double-stranded DNA. So the cybergreen um, um, can sensitively detect the double-stranded DNA, and then it goes to the minor groove and it binds there. Once the cybergreen is bound to the double-stranded DNA, it becomes fluorescent. Okay. So the cybergreen can absorb light at 497 nanometers and emits light at 520 nanometers. But the thing is that cybergreen is not specific. It's sensitive to double-stranded DNA in general, but it's not specific to a certain gene or to a certain sequence. So it can detect any double-stranded DNA in the medium. So let's say this is our sequence, or this is our DNA, and we're applying the PCR. So in the PCR, the sequence will be doubling after each cycle, as I told you previously. And let's say we have cybergreen in the medium. Then the cybergreen will detect the minor groove of the double-stranded DNA and will bind there. So at each cycle, and when the cybergreen is bound to the DNA, it becomes fluorescent. So at each cycle, we will get more fluorescence because more cybergreen will be bound to the DNA. At the end, we will get more and more fluorescence during the PCR process. And of course, the spectrofluorometer can detect this fluorescence. Yeah, the fluorescence will be coming out of the sample, and then the spectrofluorometer can detect the green color or the green fluorescence coming out of the sample. Let's talk about the advantages and disadvantages of cybergreen. So the advantages, cybergreen is easy to produce. It's a cheap product in comparison with other detectors. It's easy to handle, it's sensitive, it's very sensitive and effective to, it's very sensitive to double-stranded DNA. The only disadvantage is that cybergreen is non-specific, and so if, let's say, if there was a mistake with the primers, so if the primers don't bind specifically to the gene of interest, and so another sequence would be produced in the sample, the cybergreen cannot detect this because the cybergreen doesn't differentiate between our sequence of interest and other sequences. So we can overcome this problem by using highly specific primers. Then we can ensure that the primers will bind specifically to the gene or to the sequence of interest. Okay. Now the other type of detectors are the specific detectors, which are the detective probes. And as I told you, the most known is the Tachman. How do these detectors work? So this detector is a probe. It's a DNA probe. It's a single-stranded DNA, and it's coupled with two molecules. The first is the fluorophore. So first we have the fluorophore at the 5' end, and we have at the 3' end something we call the Kencher. What is the Kencher? The Kencher is an inhibitor of the fluorophore. Why? Because the Kencher absorbs any fluorescence coming out of the fluorophore. So as long as the fluorophore and the Kencher are bound together or are kept closely around each other, the fluorophore cannot we cannot see any fluorescence coming out of the fluorophore because the Kencher absorbs all the fluorescence coming out of the fluorophore. Now, how does this work? How can we detect the amount of DNA in our sample using this? So let's say we have our double-stranded DNA, and as you know, so let's say we have our double-stranded DNA. We want to perform PCR. So the first step of the thermocycle is the denaturation, in which we increase the temperature. So the hydrogen bonds between the nucleotides will be denaturated, and then the two strands of DNA will be separated from each other. So this is the first step of PCR. At this step, the Kencher will recognize, or not the Kencher, sorry, the Tachman will recognize the strand, our DNA strand, because it's specific. So this is exactly complementary to our gene or our strand or our sequence of interest. So this will recognize the sequence, and then the detector or the probe will bind to our strand, to our DNA strand or to our DNA sequence, because this is specific. The probe is directly complementary to the DNA, to our DNA, so it will bind to it. Now, in the second step of the thermocycle, which is the annealing step, the primers will be binding at the 3' end of the gene. This is also what I told you in the previous video. Now this going to the third step, which is the elongation step. In the elongation step, the